How to collect microbiological samples from endophytes. That is the topic of this webinar today. What are endophytes? Endophytes are microorganisms, they can be bacteria or fungi, that live inside plant tissues without causing harm to the host. These microorganisms play essential roles in plant growth, stress resistance, and disease protection. Collecting microbiological samples from endophytes is crucial for studying their biodiversity, their applications in agriculture, and potential pharmaceutical uses. It's also used in microbiological research. This guide in this video explains the proper techniques for collecting, isolating, and preserving endophytic microorganisms from plants. Number one is selecting the right plant species. The first step in collecting endophytes is choosing the appropriate plant material. Consider the following when choosing the plant material. Number one is the healthy plant tissues. Collect samples from symptom-free plants to avoid contamination with pathogens. Different plant parts. Endophytes can be found in leaves, in stems, in roots, and seeds. So, sample multiple sections. Another thing to factor in the sample collection is the environmental conditions. Plants from diverse ecosystems may host unique endophytes, so consider sampling from different locations. Number two thing you need to look at in sampling is the surface sterilization of plant samples. To isolate true endophytes, it is essential to remove surface microbes before processing the plant material. The common surface sterilization protocols involves washing, rinse plant material under running tap water to remove dust and debris, and to remove microorganisms that are in the air that have settled on the external. Number two thing you need to do is disinfection. Immerse the sample in 70% ethanol for 30 seconds. This is to disrupt microbial cells on the surface. Then number three thing you need to do is sterilization. Submerge the sample in 2 to 5% sodium hypochlorite solution for 1 to 5 minutes to kill surface bacteria and fungi. Then you need to rinse. Wash the samples 3 to 5 times with sterile distilled water to remove residual chemicals. After that, you need to dry. Blot the samples dry with sterile filter paper before processing to culturing. The next step is processing and culturing of the endophytes. After sterilization, plant samples can be processed to extract endophytes from microbiological study. Methods that can be used include direct plating method. Cut small sections of the sterilized tissue, approximately 1 cm square. Place the tissue on sterile nutrient agar for bacteria or potato agar for fungi. Incubate at 25 to 30 degrees C for fungi or 30 to 37 degrees C for bacteria. In the case of bacteria, incubate for 24 hours to 48 hours. In the case of the fungi, incubate for 3 to 5 days. Observe microbial growth from within the plant tissue that has grown on the inside of the plate and on the media. Another way to do this, instead of using the direct plant plating method, is grinding and serial dilution method. In this case, you crush the sterilized tissue in 1 to 2 ml of sterile phosphate buffered saline or distilled water using a mortar and pestle. The mortar and pestle must be sterile too. Serially dilute the extract 10 to minus 1 to 10 to minus 6 or 10 to minus 5 or 10 to minus 4 based on experience and expected growth microbial load to reduce microbial concentration. Spread 100 microliter of each dilution on suitable agar media. whether it is PDA or NA or any other suitable media you want to use. Incubate at the appropriate temperature mentioned above and monitor colony growth. The next thing is identification and preservation of the endophytes. Once endophytes are isolated, they can be further characterized through microscopic examination, and this involves ground staining for bacteria, that is lactophenol-cotton blue staining for fungi. Then you can do molecular analysis, which involve PCR, 16S RNA sequencing, and then blasting, and ITS sequencing for fungi and blasting. You can also use biochemical tests such as catalase, oxidase, and other enzymatic activities. Now, for long-term storage of the endophytes which you have isolated, you can use the following. You can maintain cultures on agar slants at 4 degrees Celsius for short-term use, or you can store them in glycerol stocks at -80 degrees C for long-term preservation. You can also use freeze drying. That means you lyophilize the cells. You freeze dry the bacterial strains for extended storage. If you are storing at 4 degrees C or at refrigeration temperature, what you do is that after like 3 months, you have to restock again and repeat the storage so that it can still be viable. The proper collection and isolation of endophytes provide valuable insights into their roles in plant health, agriculture, microbiology, and biotechnology. Using aseptic techniques, surface sterilization, and appropriate culture methods, researchers can successfully study these hidden plant microbes. Further analysis, including DNA sequencing and biochemical characterization, helps uncover their potential applications in medicine, industry, environment, and sustainable farming. This is the Master Microbiology YouTube channel. I implore you to subscribe to this channel, like this video, and share the link of this video with your family and friends. Until I come your way in the next one, please subscribe, share, and like this video and comment. This video popping up from this channel right now is the next you need to watch now. I will see you in the next one. Bye bye.